Hello, it's Mr. Burke here. I am going to give you a presentation about topic 6.5 from IB Biology about neurons and synapses. So the essential idea here is that neurons are transmitting the message while synapses are modulating the message. Have a look at the understandings for this topic, as well as the applications and skills. One fact about this topic and about neurons is that the average number of neurons in the human brain is 100 billion. There are as many neurons in the human brain as there are stars in the Milky Way. Nerves do transmit electrical signals through your body. Some examples of things that you can do because of these signals is think, move, and sense. Just as a review for previous topics that are highly involved in this topic, think about active transport, think about passive transport, and the different types of passive transport as well. So active transport is using membrane proteins and ATP to transport molecules against the concentration gradient, right? So from low to high concentration. Diffusion does not use ATP and transports molecules with the concentration gradient. Diffusion is a type of passive transport. Another type of passive transport that you're going to have to become familiar with is facilitated transport. So remember, that's the use of a membrane protein, but no use of ATP energy. And then the specific type of passive transport that we're going to be looking at is voltage-gated channels. Those are the proteins that are used for that facilitated diffusion. Okay, so what exactly is a neuron and how is it different from a nerve? Something to keep in mind is that they are different. Essentially, a nerve is a bunch of neurons condensed. So if this is a nerve, then you would have your neurons coming out, maybe branching out like this. Okay, so you'd have several of those. These are the neurons that I'm drawing. These are the dendrites. So I'm just going to draw 3 neurons with the dendrites coming out. Neurons. Okay, now let's look at the actual structure of a neuron. So as I was saying before, they're branching out at the end. Okay, and these are called the dendrites. And then you have the cell body, also known as the soma. Cell body. That would be around this area. Cell body. soma. Okay, and then that circular thing, since this is a cell, this is just a very specialized type of cell, that's why it looks a lot different. This is the nucleus. Along here, the longer part that transmits the signal, the electrical impulse, is called the axon. Okay, and then towards the end of the neuron, you have another sort of branching part known as the axon terminal. So you can remember that because it's the end of the axon, the terminal end of the axon. Okay, and then some other things that you will be learning about in relation to these neurons is there's some covering, a fatty covering that is all along the axon here, known as the myelin, specifically the myelin sheath. Okay, and then you have these cells within the myelin that produce the myelin that are called the Schwann cells. The myelin helps to transmit the signal to be faster as it moves along the axon. It's known, this action that happens with the signal transmitting faster due to the myelin is called saltatory conduction. Okay, and then there are these spaces between the myelin, so unmyelinated parts of the neuron, that we call the nodes of Ranvier. Okay, and so the important pieces from this, the most important pieces that you need to know about are dendrites, cell body, axon, axon terminal. Okay, and then as you progress on, you'll learn more about that saltatory conduction and how myelin is connected to the saltatory conduction and the Schwann cells and the nodes of Ranvier. In terms of what do each of these parts do, what is their function, the dendrite's job is to receive information from another neuron or possibly some sensory cells. Okay, it's going to be receiving the information and the stimulus, and if the stimulus is big enough, then an action potential will start, and that action potential will be passed along the axon to the axon terminal. And then from the axon terminal to where? To another neuron, or possibly an effector cell, like your muscles. So here's a quick example just showing how there could be a stimulus that is received by some receptor cells, such as the cells in your eyes. Okay, in this case, the eye is looking at a glass of water. There's a sensory neuron that would pick up the signal from the receptor cells, okay, on your sensory organs, and then the sensory neuron is going to transmit that cell, or that signal, to a relay neuron that would be in your brain. Your brain processes that and sends the signal further to a motor neuron. The motor neuron would be connected to effector cells that could be, for example, in your muscles, and the response would be for the muscle to contract and you to drink the water. So as I was saying before, you do need a good understanding of the different types of membrane transport, specifically facilitated transport, which is going to be with these voltage-gated sodium channels and the voltage-gated potassium channel over here. So the main idea with these channels is they are channels, right? Which means channels are always going to be with passive diffusion. A pump, however, this is the sodium-potassium pump, that's going to require ATP energy, so it's going to be active transport against the concentration gradient. These voltage-gated sodium and potassium channels will be with the concentration gradient, and it does not require energy, even though it's slightly deceiving because they are voltage-gated channels. Okay, so what does voltage-gated mean? Well, I'm going to first just draw a little lightning bolt so that you remember. It does need a specific voltage or membrane potential in order for them to open. Now, what is membrane potential? That's what we're about to talk about. So, if this is the neuron membrane right here, this would be outside the neuron. This is inside the neuron on this side. And we're going to discuss something called membrane potential. So essentially, membrane potential is that difference in charges between the outside of the neuron and the inside of the neuron. What do I mean by that? Well, there are all of these ions of sodium and potassium that are involved here that create differences in charges from the inside to the outside of the neuron. So the way that it starts off is outside the neuron, it's more positive. So I will go ahead and write more positive. Okay? Inside the neuron, more negative. Okay? This is what causes a membrane potential because there's an electrical gradient. So just like there can be a chemical gradient, there can also be an electrical gradient. And in this case, the electrical gradient is produced by having more sodium on the outside of the neuron and more potassium on the inside of the neuron. It causes there to be a resting membrane potential of, so resting membrane potential equals negative 70 millivolts. Okay, so then we've got over here the sodium channel, right? So it is voltage gated, meaning it's only going to open when there's a specific membrane potential, and that is not negative 70 millivolts. So as I said before, though, there is this difference from the inside to the outside of the ions, the sodium and potassium, that causes this resting membrane potential, this initial resting membrane potential, to be created. So what is that difference? Well, there's way more sodium ions outside of the neuron than inside. So I'm just going to draw a bunch of sodium ions all throughout here. Okay? It is more positive on the outside because there are more positively charged sodium ions on the outside or in the outside of the neuron membrane. Okay, and then for the potassium, there are fewer potassium ions, which means a less positive charge on the inside okay, less positive charge on the inside than outside. And because it is a less positive charge on the inside than the outside, that creates this resting membrane potential of negative 70 millivolts. It's a net charge of negative 70. So you might be wondering, what is it that actually causes this resting membrane potential to be negative 70 millivolts? And the answer lies in this sodium-potassium pump. So the sodium-potassium pump helps to restore that resting membrane potential. That's what we want. We want that charge. We want the more negative charge, negative 70 millivolts, inside the neuron than outside, because that's what it has to be at while it's not firing. Okay? When the neuron is firing, that's when an action potential will be, or the signal, the electrical signal, will be transmitted along the axon. So when the signal's not being transmitted, the resting membrane potential is negative 70 millivolts, and the thing that helps to maintain that is the sodium-potassium pump, which, as I said, does require energy. Why does it require energy? Because it is pumping against the concentration gradient. Okay, so the ATP goes in and is used and turns into ADP, of course. Okay, now let's talk more about this action potential and how that actually comes about and how that ends up opening the voltage-gated sodium channel and eventually the voltage-gated potassium channel, and then how the sodium-potassium pump once again restores that resting membrane potential. So here's a zoomed out view of what I was showing you before with your voltage-gated sodium channel, voltage-gated potassium channel, and then the sodium-potassium pump. Remember that it's more positive outside the neuron, more negative inside the neuron. These two blue lines are the neuron membranes. So this is one of the sides of the neuron membrane that these channels, the voltage-gated channels, are in, and the potassium pump. So what happens here with action potential? That's what we're going to talk about. Remember, the membrane potential actually starts at negative 70. Do need to make a quick correction here. This should be negative 55 over there. Okay, but for negative 70, resting potential is negative 70 millivolts, and the thing that's restoring that always is the sodium-potassium pump. But what actually ends up opening these voltage-gated channels? So first, there's a stimulus that is received, right? And as the stimulus is received, the resting potential is going to move over on my graph over here from negative 70. It's going to increase, and if the stimulus is great enough and changes the membrane potential to being past the threshold, then what happens at that point is something called depolarization. Okay, so if membrane potential greater than threshold, depolarization begins. Okay, and sodium channel opens. Okay, so when the sodium channel's open, the sodium is going to diffuse across because we know that this is passive transport. Okay, it opens because it's voltage gated, and the voltage that allows it to open is just past the threshold. So then the sodium starts moving in. What do you think is going to happen to the membrane potential if positive ions, more positive ions, are moving into the membrane? It should become more positive. And that is what depolarization is. So we're going to show that on the graph. It's going to keep getting more and more positive past 0 and all the way to positive 30. That's a 30 over there. All the way to positive 30. Depolarization. Okay, and then once we're at positive 30, that is the voltage that allows the potassium channel to open. Now, what's going to happen when we're removing positive ions from the inside of the membrane? If you said that the membrane potential will get more negative, you would be correct. So repolarization happens when membrane potential reaches 30 millivolts, okay, and the sodium channels open. Okay, so we're going to then show, because remember we said it gets more negative when the potassium channels open. Did I just say sodium channels? I think I did. That's when the potassium channels open. Okay, so we're going to show that on our graph over here. It's getting more and more negative, gets below the threshold, below even the resting potential, and we call that hyperpolarization because it's getting really, really negative inside the membrane because of all the potassiums leaving. And then there's this period called the refractory period where it takes some time to get back to the resting potential. Okay, so I'm going to just write repolarization. Okay, this area from here to here is the refractory period. Okay, and what is the thing that restores the resting potential? That's going to be the sodium-potassium pump. So just write during refractory period, sodium, potassium pump restores resting membrane potential. Okay, and there you have it. That is an action potential that does get propagated along the neuron. So it's like a chain reaction where as you're moving along the neuron, along the axon of the neuron, the signal and the depolarization will just be propagated and move along the axon until you reach the axon terminal. This slide is a really nice overview slide if you need a quick review of what happens during action potential. Okay, it has that similar graph that I was using. Okay, the first part, remember, is the resting potential has to be maintained as the neuron is not firing. If it's not receiving a stimulus, then it's going to keep that same resting potential at -70. And it's the sodium-potassium pump that does that through active transport. Okay? Depolarization, which, by the way, I did not show that happening in this image over here, so let's go ahead and draw that happening as well. Here, you've got those sodiums actually end up, the sodiums that are inside the cell, which are over here, the sodiums inside the cell, get transported back outside of the cell with that sodium-potassium pump. Okay, and then the potassiums are going to be brought actively back into the cell to restore that resting membrane potential of -70, where we have more sodiums outside, more potassiums inside, and in fact that sodium-potassium pump is taking, for every 3 sodiums that it takes out of the neuron, it brings 2 potassiums back in. And so the idea behind that is it has to maintain that idea of it being more positive outside than inside so that there's a net -70 charge. Okay, so if we move back over here with action potential, as a review, remember the resting potential is maintained by the sodium-potassium pump at -70. Then depolarization is going to happen if a stimulus is received and the membrane potential goes above the threshold of -55. Okay, then as it gets more and more positive and all the way up to +30, that's when the voltage-gated sodium channels close and the voltage-gated potassium channels open. That's when we're repolarizing and getting more negative. And then it gets very, very negative, hyperpolarized, and there's a refractory period, which this refractory period is actually important for making sure that the signal does not move backwards, so it doesn't move in the opposite direction that we want it to move in. I'll talk a little bit about that later. And then the resting potential is maintained once again by taking out 3 sodiums and bringing in 2 potassiums for every 3 sodiums. And that is an active process against the concentration gradient. So if we look at this slide, this shows that propagation and the direction of the action potential. It is moving from the dendrites to the axon terminal, and as it's moving along, subsequent sections of the axon become depolarized, while the previous part of the axon, where the signal once was, is now repolarized, and then eventually goes back to resting. Example of a question that you could see on an IB exam. So you can pause here and try to answer it, and then I will show the mark scheme. Here is the sample mark scheme for that question.